Okay, good morning. So today is our actual last lecture of new material for the semester. So we're wrapping up our discussion about clinical treatment of cancer. We're going to talk a little bit more about the rational design of different types of drugs. And this is a nice way to put together a lot of the concepts that we've been talking about over the course of the last 15 weeks or so. So as a reminder, we will have one more quiz. So we'll have one more quiz that covers this last chapter of material, and that quiz will be due next week as normal. This Friday is another article share day. So the article share assignment is available in La Lima for you to submit by tonight. And then on Friday, we'll have our last article share day. On Monday, I will use our class time to share with you some of the nature of the work that I've done. And as I mentioned to you previously, there's two sort of areas of research that I'm involved in that I think students particularly are interested in. I will set up a poll in La Lima to allow you to decide which of the two topics you want to hear about on Monday. So that means today's class is the last new material that you're responsible for the class. Monday's class time will just be me talking about the work that I do, because I think it's nice. I think many of you are curious about why does Dr. Shen teach cancer biology, right? So I'll share with you the work that I do. And then next Wednesday will be a review for our third exam. And then the following week on May 10th, at the scheduled final exam time for our class, we will have our third exam. All right? Okay. So let's go ahead and wrap things up talking about drug design and also what are clinical trials? How do clinical trials work? So we left off last time with this slide here. Last time we had talked about different types of drugs, things that work very systemically, for example, like the taxanes or vincristines that target things like microtubule formation in cells, preventing things from spindle fibers during mitosis to form. And that's a very effective way to stop cells from dividing. Right? We also talked about compounds like cisplatin, platinum-based drugs that can easily diffuse into cells right through the membrane, because remember, cisplatin is just a platinum ion with like 2 amine groups and 2 chloride groups. So it's kind of amazing to think that this little molecule itself is a very potent therapeutic. But the way it works is it gets into your nucleus of your cell and starts forming adducts with DNA. And as a result, the cell cannot fix all of these platinum adducts, and the cell should then undergo apoptosis. In the case of a cancer cell, right, this cancer cell simply can't divide anymore because its DNA is all cross-linked. In both of those cases, when we're talking about these sort of systemic drugs, I think it's quite evident to you that these types of drugs cannot discriminate between what is a cancer cell that should be targeted versus a normal healthy body cell. And consider, right, that even in the body of a patient who's suffering from a late stage solid tumor, still the majority of cells in the body are normal body cells. So if you have these compounds like the taxanes or platinum compounds flowing through their system, right, these are highly toxic drugs. That is why I think you're all familiar with many of the really terrible side effects of chemotherapeutics. Things like hair falling out, things like the patient unable to eat any food, right, because their gastrointestinal tract is all messed up. Why is that? Because these drugs interfere, right, with the ability of cells to properly go through the cell cycle and divide. So the cells in the body that are hit the hardest wouldn't necessarily be something like a skin cell that doesn't really care about dividing anyhow, right? Like a keratinocyte in your skin, right? But the cells in tissues that do rapidly divide, things like the cells in your hair follicles that are constantly functioning to produce more keratin, for example, for your hair, or the epithelial cells of your GI tract, right, which constantly have to be replenished. So there are cells in there that are dividing. Those types of tissues are hit the hardest, and that's why you get those types of awful chemotoxic side effects of these systemic drugs. In many ways, then, right, the treatment of cancers with systemic drugs is kind of like a game of chicken, right? You're trying to get the biggest dose possible to try to rapidly target the tumor cells and attack the tumor cells, while at the same time trying to mitigate, right, or balance out how much other off-target toxicity is acceptable, right? So it's like you give the patient a high dose of these drugs, you are essentially killing them, right? So how do you play that game of chicken of you're killing them, but thank goodness the tumor was attacked, and so now you can take and back off on the drugs, right? So that sounds kind of barbaric, right? And it really is. A lot of these compounds, these drugs that are used, that have been used for the past 50 years or so, are very barbaric in nature because they're systemic, right? They don't really go after a particular rationally chosen target. So that's why rational drug design is so important and such a hot area of research. Remember, we said rational drug design means you start off with a target in mind, right? And then you try to understand the biology of that target so that you can then design or come up with some sort of molecule that can specifically interact or modulate the activity of your target in the way you want. And last time we ended with a discussion that when you're going to approach drug discovery sort of from a de novo or brand new perspective, right, designing something new, you have to deal with things like the feasibility of the target. What is your target? How does it work? The specificity of your drug, right? If it targets your particular molecule of interest, there's probably a good chance that it might target other molecules in the cell as well. Bioavailability, that has to do with pharmacokinetics, how long the drug can actually last in the body before the body just gets rid of it as part of normally detoxifying our tissues, and overall efficacy in vivo, right? In an animal model and ultimately in a human body, how does this work compared to the excitement that you saw maybe in a plastic dish when you tested it out in the lab? So let's go ahead and explore these a little bit more in depth. First off is feasibility, right? How does one go about finding a particular target that you even want to go ahead and start designing a drug against? So one way that we are able to now use the power of computers to help us look for targets has to do, again, with these types of microarray, really high throughput analyses. Right? We've seen something like this before, where different columns and different rows mean different things. Here's an example where they're looking at breast cancers, right, and trying to identify certain maybe families or groups of genes that might be very useful, not only in identifying something about the etiology of the breast cancer, but also providing some insight as to maybe what genes or what proteins made from those genes might be good molecular targets. So let's look at this. Every row in this matrix here is a different tumor taken from a different patient. So you have 295 tumors. Each row is one of these tumors. Right? Each column is one of 70 different genes that there was some a priori or beforehand knowledge or belief that this gene might be relevant in breast cancer. Okay? And so then in these 295 tumors from 295 patients, they went ahead and looked at the expression levels of these genes and just said if the expression level was more or less equal, maybe they would give it a black sort of color, right? Whereas if the gene looked like it was underexpressed in the tumor, right, it would be more green colored on this chart. And if that gene looks like it's overexpressed, either through amplification or some other type of overexpression, the more red it would be on this. Right? So you can imagine then if you randomly pick any one of these rows, so for example, let's just pick the row that's at like 250 right here, tumor number 250, and you were to go across, right? So you would have sort of this like almost hodgepodge assortment of some genes are red, some are green, some don't change at all, right? The computer can then organize all of this data in a way that lines up all of these rows, right, and all of the columns to be able to generate a pattern. And that's the pattern that you see here, right? And this is really cool because obviously, human beings, we could do this by eye, right? But it would take forever. It would be so methodical and boring, but a computer can just do it. And so by lining it up this way, you find, right, that you have a group of patients down here that have overexpression of this set of genes, right? And you have a group of patients up here that have overexpression of these genes. Right? And what's interesting is you can see it's actually, contrasting, right? So if you have high levels of these genes expressed in your tumors, right, you typically find that you have lower levels of these genes over here and vice versa. What they then do is they sort of set a threshold here, right? And let's just say they arbitrarily put this dotted line in the middle, right? And then they say this represents one signature, and then everything on the bottom represents sort of a different signature or a different sort of fingerprint. What they find is if they group those people and follow them in a Kaplan-Meier plot, right, the upper group turns out to be a very good signature. You can see that these patients survive 10 years out, whereas patients who were sort of part of the lower signature have a much poorer prognosis. Right? So this is very useful. So that means you can already identify certain genes in this group of 70 genes that might be suitable targets to give you some idea of whether or not this patient's going to have a good or poor prognosis. Right? And based upon that, you might focus on any one of those genes as a potential target for a therapeutic. Why is that so important, using this type of technique? Let's look at an example of this type of cancer here. So this is, this is diffuse large B-cell lymphomas. Okay. So lymphomas are solid tissues formed by B cells or precursors of B cells that as they're dividing in the bone marrow, they lose control of dividing and they become these solid tumors. So there's three main disease subtypes of this diffuse large B cell lymphoma that patients present with in the clinic. And they are called primary mediastinal, germinal center, or activated B cell-like. You don't have to know the names of these, right? But I think if you follow what this example is trying to teach you, I think you'll see why some of these techniques are so important. The problem is when a pathologist has a biopsy from a patient that presents in the clinic with, they think, oh, it's B-cell lymphoma, and the pathologist looks under the scope, this is what they see. All three types of this disease look very similar. You cannot tell the difference just by looking under the scope, right? But this is a problem, because even though all three of these types look very similar morphologically in terms of the cells and the tissues, right, the patient prognosis is very different depending on what type of disease they have. You can see patients who have this PMBL type, right, have a better prognosis going forward in this Kaplan-Meier plot compared to the other types, the GCB, and then the worst prognosis is if the patient ends up having ABC, or an activated B cell-like. So right off the bat, you can understand that there's an urgency to be able to somehow identify what types of intracellular signaling pathways distinguish these three different closely related diseases, because that will give you that valuable knowledge about what things you might be able to target to treat these patients based upon which subtype of the disease they have. And certainly in the cases of patients who have ABC type, you don't have a lot of time to waste, right, guessing on different drug treatments and then realizing later, okay, that didn't work because I guess that doesn't work for this particular subtype that they had, but we didn't even know that they had this subtype from the start. So let's look at how you can use these types of microarray data, like we saw with the breast cancer, to look at these lymphomas. So again, similar type of heat map matrix here generated. In this case, on the rows this time, so it's kind of flipped over on its side, the rows are different representative genes, right, that are being assessed for expression level. Either low expression level green, regular normal expression level would be black, and then highly overexpressed would be more red, right? And then what they've done is each of these rows now, right, is oh no, sorry, not each of these rows. Each one of these columns now going down would be a different patient, right? A different patient who at the time of presenting in the clinic, right? This is what the pathologist saw under the scope. So they believe, yeah, it's one of these types of B-cell lymphoma, but what is it, right? So they have all these columns or a different patient being evaluated, their tumor cells are being evaluated for expression levels. Right? So if we just left it at that, right, you might imagine since these three different types of lymphomas have very different sort of behaviors, right, probably very different cellular pathways, probably even when the computer tried to sort it out, it'd be a big mess if you just left it all as being collected as B-cell lymphoma, right? But now if you split it out, right, into different sort of subtypes, you can see the PMBL, the GCB, and the ABC, right? If you further kind of divide this this matrix into three bigger sort of column groups, something amazing happens, right? You can really see now the subtype specific signature of gene expression in these different types of lymphomas, right? So the PMBL lymphomas typically have highest expression levels of these groups of genes, right? These first top two groups, but low levels of these two bottom groups of genes. The GCB has high expression level of these two middle groups, and the ABC has high expression of only this bottom group of genes, and then the other ones are either low or it's a little bit more ambiguous up here. Okay, so if you break this down, you can start making some data or getting some data from this, right, and analyzing that certain types of mutational events like BCL2 translocations, right, seem to be happening at higher frequency in GCB versus PMBL and not at all detected in the ABC subtype. Or things like activation of different types of oncogenic, potentially oncogenic pathways, like the NF-kappa B transcription factor, right, which is a very important transcription factor for many genes. You can see in PMBL and ABC, they're able to detect either overexpressed or constitutively active NF-kappa B pathway, whereas in GCB type, you don't see that. So that provides you with a lot of useful information because you may already have drugs available that target these different things that maybe previously, right, have not really been used for something like lymphoma, but this might give you motivation, right, to try to investigate whether these drugs might actually work for certain of these different subtypes. For example, let's look at that NF-kappa B activation, right? So 2 out of the 3 subtypes of these large B-cell lymphomas have overactive NF-kappa B, right? And this third one does not. So if you treat patients then with a drug, this is a I-kappa B kinase inhibitor that impacts or shuts down NF-kappa B, right? What you find is, sure enough, right, this kappa B drug, I-kappa B inhibitor, is effective in killing off. So this data now is not a Kaplan-Meier plot. This is actually in vitro cell culture data, where you're growing these different types of lymphoma cells in a dish, and you're treating them with the drugs and seeing how well the drugs actually lead to the killing of these cancer cells. And you can see the drug treatment is effective for the PMBL and the ABC type, but not the GCB type, right? Which makes a lot of sense when you realize that the NF-kappa B pathway itself only seems relevant for the ABC and PMBL. Right? So you can see by having this type of data, you really, really have an advantage in being able to be smart in choosing specific drugs that target specific pathways, because you're able to see that these pathways seem to be relevant in the cancer that you're interested in. All right. So this summarizes kind of this concept of choosing a target, because let's look at a cartoon that should be somewhat familiar to all of us by now. All right, this is sort of a, it feels like a long time ago in the semester when we first learned about these types of signaling pathways that have to do with things like EGF receptors. So here's HER2 at the top. Right, this is the HER2 new receptor. And remember, it's a receptor tyrosine kinase. So it acts by doing things like recruiting different adapter proteins like GRAB and SOS that can then activate RAS, right? It can recruit SRC, and then it can activate other things like Janus kinases, and all these things end up sort of overlapping and turning on different pathways, like the PI3 AKT pathway, and these different mitogen, or these different MAPK pathways. Right? So all of these pink sort of indications here are showing you that each one of these different proteins in these pathway serves or can serve as a target, right, for a particular therapeutic drug to go after and interfere with just that component. And indeed, all of these pink things show you that these are different groups of drugs that we currently have available. And it's kind of neat to see it this way, because that allows you to see, right, all these different types of drugs designed against different components of the pathway. These are the different components that they go after. Right? Everything from PI3K inhibitors to SRC inhibitors to RAS inhibitors to inhibitors of the receptor tyrosine kinase itself. Right? So the more you know about the pathways that link things like oncogenic signals ultimately to cell phenotypes, things like cell cycle progression or survival or proliferation, the more opportunities you have to try to target and disrupt these pathways. Okay, great. So let's say, there's an abundance of these different targets then, right? So you say, hey, that's great. If I find a protein that's part of a pathway, then I'll go ahead and design a drug against that, right? But this is where a particular problem comes in. And this has to do with specificity. All right, what do I mean by specificity? Because remember, many of these proteins have very conserved evolutionary domains in them, right? Things like Sark homology domains or kinase domains, things like that, that Mother Nature along the way in evolution stumbled across a particular folded peptide region, of amino acids. And this works really well as like something that, transfers a phosphate group. And so proteins that have this as part of their gene, right, they end up making this part of the protein, and they are kinases, right? So as a result, right, because all of these things are shared through evolution, oftentimes the shapes of these functional regions of these proteins are very similar to some other protein that might be closely related, but is involved in a different pathway. To illustrate that, let's look at this. Here are a bunch of kinases, right? So here are 9 different kinases, right? So these are all, and these are just the kinase domains of these proteins, right? So the rest of these proteins are really big, but they're just showing you the ribbon diagram of the kinase domain part, the part that actually takes care of transferring a phosphate group onto a substrate target. In blue are serine threonine kinases, some examples of them. Remember, like 99 point, I don't know what it is, 0.9% or whatever of kinase activity in the cell involves phosphates being transferred onto serine and threonine kinases. Right? In our studies of cancer, we learned that tyrosine kinases, right, are sort of reserved for those very special cell functions that involve things like cell cycle and proliferation, right? Because those are like the VIP kinases, because those are pathways that you don't want to sort of mess around with, right? You ultimately need tight control over things like cell proliferation to avoid cancer. Right? So that's why we have some of our familiar friends here. So here, for example, here's a cyclin-dependent kinase, CDK2, the kinase domain. Here's our friend SRC down here, right? This is the tyrosine kinase domain of SRC. And what you notice, hopefully, when you look at these 9 examples of kinase domains, is they all look the same, right? Because again, they all evolved, hundreds of millions of years ago from some ancestral gene, right? That just through random mutations came upon this right shape that's very good at, when you make it into a protein, at transferring phosphate groups. Right? So this is the problem. This figure on the right really illustrates this problem. If you have 2 different kinase domains from 2 different, very different kinase proteins, and you overlap them, right? Sort of like taking 2 of them and making like a composite image, and where their position is close to being the same, right, you make that gray. Right? Where their shape is different at all, the more different they are, you make those positions green. Right? And what do you see? You see, for all intents and purposes, right, that these kinase domains, right, are nearly identical. So if you were to design a drug that targets the particular 3-dimensional shape of this kinase domain for one particular protein, for example, SRC, there's a really good chance that in the body, it's also going to interfere with a number of other kinases, right, that might actually mess up things in the cell that you don't want to mess up with. So that's why carefully understanding protein structure and being able to iteratively, iteratively means what? You take something and you sort of carefully build upon it in little steps. We're talking about little drug shapes here, right? Iterative kind of medicinal chemistry to add things to the shape of a drug can really improve specificity. Here's an example. Here is the kinase domain of the BCR-ABL oncogene. Right? So the BCR-ABL oncogene, remember, it's this weird chimeric protein, and it has kinase activity. So this is a kinase domain, right? So this green thing looks a lot like the rainbow-colored stuff on the previous slide. And here's a drug that's called Gleevec. So Gleevec, or imatinib, is a very, very popular drug for treating myelogenous leukemia because it targets the BCR-ABL oncogene. So here you can see in blue is the drug in here, all right? And that's how it fits into this kinase domain and prevents the kinase domain from working. Here's the drug on the left. This is Gleevec. Right? Really interesting shape, right? It's like a series of rings, right, that are strung together. Many of these drugs are like this because when they are rings, the drug tends to be more planar, right? And so if there's particular surfaces or grooves in a 3-dimensional protein that you're trying to target, oftentimes these planar structures of small compounds fit in there better, sort of like a key going into a lock. So what they're showing here is that Gleevec is the result of starting from a structure like this, right, and carefully modifying it, putting things like an oxygen on this position here and making sure that there's a nitrogen at this position here. When you look at this blown up zoomed in image, so here's the Gleevec in here, you can see this is where the nitrogen is and where the oxygen is, right? And the green surrounding it would be all the amino acid side chains of the BCR-ABL oncogene, right? So you can see that these positions in the drug are designed or put into this particular position because they're put in the right place then to interact with, for example, this asparagine here or this isoleucine here, right? That the kinase domain of BCR-ABL might have, right? But maybe the kinase domains of other kinases don't. So as a result, Gleevec binds very tightly to BCR-ABL, right? And not as tight to other kinases. So if you see this data here, what we're looking at is some cellular data. So this is in vitro stuff where we're treating cells with this Gleevec and asking, right? The different kinase pathways that take place in the cells, which ones get inhibited more by the presence of this Gleevec drug compared to others. And you can see very clearly that a number of these kinase pathways, right, do get affected by Gleevec, because on the y-axis, this is percent of inhibition, right? How strongly are you inhibiting this particular kinase pathway with Gleevec? You can see all of them, there is some limited off-target effect, right? But because of the specific nature of the Gleevec, by designing it with that nitrogen and that oxygen just in the right place to take advantage of what the shape of the BCR-ABL's kinase domain is with the asparagine and the isoleucine, you can see in light blue, right? It's the c-ABL kinase domain that gets impacted the hardest by the Gleevec, right? So isn't that cool? I think that's really cool, right? So starting with a molecular scaffold structure that might have, inhibitory effects on, let's say, kinases, you're able to tweak that structure by adding particular little groups to it so that it gets really customized to bind your desired target much tighter. And what does that mean? The more tighter the drug targets your particular molecule of interest, the lower dose you can get away with using, right? In order to more specifically go after a particular pathway and spare the cell, off-target toxicity. All right. So when you're first looking at these drugs, obviously you're first doing experiments with these drugs using cell lines in the lab. Right? So let's see how something like this can be determined. So let's say you wanted to study Gleevec and you wanted to understand, how does Gleevec work? Is it toxic to the cell? Does it work to actually stop these lymphocytes? So here are some healthy, normal pre-B lymphocytes. So these are sort of like stem cell-like pre-B cells that are growing. Right? And these B cells, they need interleukin-3 as a signal to proliferate. So in your experiment, you can treat one dish of these cells with IL-3, and sure enough, these healthy pre-B lymphocytes, they begin to divide. Right? If you actually remove all the IL-3 from the media, right, so there's like zero IL-3 in there at all, these cells die because they don't receive that survival signal. Right? So that's nice. That already shows you the sensitivity of these cells to the natural normal signal that they need in order to grow. And without that IL-3, they don't grow. Importantly, as a control, you can add IL-3 to the media, and you can also add Gleevec to it, just to see what happens to the cells. And what you find is that the cells still grow. Right? So what this shows is that Gleevec is not just sort of like intrinsically toxic. There's not some weird global toxicity effect of the Gleevec. Right? So then what you do is you turn these cells into sort of like cancer. Right? So you go ahead and you put in the BCR-ABL plasmid. Right? So now these cells have been transfected with BCR-ABL. So now they have this strong oncogene in them. And then these cells, if you now remove the IL-3, remember originally these healthy cells, if you remove the IL-3, they'll just die because they depend upon the IL-3. But these transformed cells with the BCR-ABL, if you remove the IL-3, oh boy, they keep going, right? They keep growing. Therefore, it shows that the BCR-ABL can make them IL-3 independent. You can also show that if you keep the IL-3 in there and you add Gleevec to them, right, then even if the BCR-ABL part gets stopped by the Gleevec, the fact that you're giving them IL-3 keeps them growing. Right? So in this step, what you're looking for is drug specificity, right? Because you know that this drug specifically is supposed to target the BCR-ABL. Right? So if BCR-ABL is in there and you give it Gleevec, right, but you're also keeping these cells going by giving them IL-3, which is what they naturally want, you don't see any cell death, right? So that's nice. It shows you the Gleevec is probably pretty specific to the BCR-ABL. But the thing you really want to see is what? In these cells over here that don't have IL-3 keeping them going, and they're going just because of BCR-ABL, now if you give them the Gleevec, that takes the BCR-ABL out of the picture, right? Now the cells die. And this then shows you how effective the drug is in being able to kill the cells that were becoming IL-3 independent. Right? So these three sort of steps then in this pretty simple example is the basis of how you would go about studying a new drug in the lab. So you would have to choose a cell line to work with, right? Because if you're going to work with cells in a dish, you have to have a cell line that represents a well-established model, right? That if you're going to do experiments with and you publish on it, people will actually like know what you're talking about in terms of what cells you're using, right? These are not just some random cells that you came up with, right? Because who knows what they are. So many cancer research or drug research experiments are done using a particular selection of cell lines. The National Cancer Institute has this library of cell lines called NCI 60. So it's like 60 different well-established cancer cell lines. And this panel of cell lines, oncologists in the lab, choose from this catalog of cell lines. You choose something that sort of closely matches your particular disease of interest, right? So for example, if you're studying prostate cancer, like I do, right, most of my work had to do with 3 or 4 different prostate cancer cell lines. Right? Many of you have encountered the names of some of these cell lines in the articles that you've reviewed. Right? So in breast cancer, things like MCF7 is a very popular breast cancer cell line. Right? Okay. Importantly, though, where did these cell lines come from? These cell lines came years ago from some patients' cells, right, that were harvested, maybe part of biopsy, and were introduced into the lab to see if they would grow, right? So oftentimes you'd have many different cells taken from different patients, and researchers try to grow them in a dish in the cell. And what do they find? They find that most of them won't grow very well, right? We've spent a lot of time this semester appreciating the fact that even though a cancer cell in the body is sort of like a rebel cell, right? Especially like an epithelial cancer or carcinoma, and no longer properly listens to signals from the stroma. But what? We learned that it still needs to have the support of stromal tissues, things like that, in order to actually be okay growing, right? So if you just remove these carcinoma cells and put them in dishes, in media, right, you wouldn't be surprised if many of them don't grow, right? But every so often, you would encounter a particular patient with a particular tumor who you took those cells from, you put them in the cell culture, and whoa, these cells like just take off, right? So not surprisingly then, these very famous, well-established cell lines tend to represent original tumors that were super aggressive, right? Maybe more so than your typical tumor. So if you look at these here, so these is some data. So this is like a Kaplan-Meier plot. It's kind of a weird one though, because this represents cells that were taken from different patients and then tried to introduce these cells into cell culture to see if they could grow and establish a new cell line. And what you find is if you look at how these cells do in terms of being able to establish a cell line in a dish in the lab, and you compare them back to how did the patient do in terms of their overall health and survival, right? On the x-axis is actually survival of the patient, and on y-axis is the survival, right? That part's sort of your traditional Kaplan-Meier, but the colored lines show you from which patients did you successfully get cell lines from. And not surprisingly, the cell lines, right, these pink ones, came from the patients that had super aggressive tumors, and therefore the patients themselves did much worse, right? But yet you were able to get cell lines from them. Whereas the patients who did better, the blue line, right, there was a harder time establishing cell lines from them because their tumors weren't super crazy aggressive. So what does that mean? If you're using then these well-established cell lines that originally came from super aggressive tumors, how can this might bias, right, the results you have when working with these cells in the lab, in vitro? Because in many ways, maybe the findings that you get from your experiments might only reflect what you see in super aggressive cancers, and that might not necessarily be the type of cancer you're hoping to target, right? Maybe the drug you're working on, you're trying to use it as like a first line sort of treatment for patients who have low grade cancer, right? Maybe these cell lines aren't the best choice. So once you establish that something works in a cell line, you have to see if it works in a living tissue system. All right, so this is moving now from in vitro to in vivo. So not surprisingly, certain animal models are very popular to use. Most popular probably for cancer research are different inbred mouse strains. What do we mean by inbred? These are different strains of mice that years ago, right, were intentionally inbred within their specific population in order to create a genetically homogeneous line of mice. Right? Because one thing we've studied, right, is that cancer is very dependent upon the person's immune system and immune profile. Right? So you don't want to have a situation where, okay, I'm going to do an experiment in a dozen mice, and these dozen mice are just randomly 12 different mice that have very different genetic backgrounds, because that might have a big impact on the data, right? It might be very hard to actually see something clearly. So you want to use, let's say, a population of mice. The 12 mice all have essentially the same genetics, right? So some famous mouse lines are the bulb C mice, these white mice, very classic lab mouse. I did a lot of work with these bulb C mice. I like them because they're very docile, right? When you open up the cage, they're sort of like happy to see you, right? And they're really nice. They don't bite. Right? C57 Black 6 is another popular one, but this mouse is like the opposite. It's like super aggressive. I remember opening up the little cages of mice, and then the brown or black 6 mice inside would all like rear up on their hind legs, and I swear they'd start like hissing at me, right? And they bite, right? I remember being in the lab and having some of my coworkers, they start shouting and they're waving their hand around, and dangling from their gloved finger is a black 6 mouse, like thrashing around, right? Then there's immunocompromised mice, right? Like these nude mice, right? Who just have a defective immune system. Yeah, okay, so there's no such thing as a karaoke mouse. I just put that in there just to see if students pay attention to the slide or not, right? So these different mice have different attributes, right, that make them good for research. The karaoke mouse, you give it a little alcohol and then it really loosens up. And okay, well, anyhow, right? So let's move on because we're running out of time. So let's think about this. What happens, the importance of cell lines versus in vivo tissue environments for studying cancer. If you were to take, let's say, cancer cells from a human being, let's say from a lung tumor, okay, and you were to look at it under a microscope, here, well, the cartoon shows it's coming from the lung, but these pictures are prostate tumor and colon tumors. So these are actual surgical specimens of how these tumors look in vivo in a human body, right? If you were to take some of these cells from the prostate tumor or the colon tumor from a human and inject them into one of these receptive mouse strains, and then the tumor grows, and you look at what it looks like, you can see already just by going into a different type of animal, right, the tumor begins to change its morphology a little bit. Not a million miles away, right? Because I think it's still pretty clear that the engrafted patient specimen in the mouse for prostate tumor still looks more like a human prostate tumor than, let's say, a human colon tumor. And likewise, the engrafted colon tumor in the mouse still looks more like a human colon tumor, right? But the problem is this: you cannot always rely upon just having patient tissue to inject into your experimental mice, right? I mean, that's just not practical. You can't say like, okay, I have 20 mice, so I need 20 fresh tissue samples from a patient or from different patients, and you get them and you do your experiment. And after your experiment's done, you're like, all right, I have to repeat this. So I got to go back and open up those patients again and get more tissue, right? That's obviously not practical. That's why we use cell lines. because cell lines serve as a nice intermediate, right? These were originally human cells that were introduced into culture, and now, right, you can just order them from a catalog. You can grow them up, you can freeze them. When you need to have some more, you pull them out of the freezer, you thaw them out, you grow them, and then you can inject these into your animal models. But the problem is this: look at what these look like now when they've gone through this extra step of being a cell line and then injected into the mouse, right? You see now the appearance of the cancer looks very different, right? So the question is, if you go ahead and do all your experiments on these sorts of tumors here, how accurately do they really reflect the biology of the cancer in the original human tumors, which are the things that you want to target, right? That's what you're really interested in designing drugs against. All right, so a couple more slides, and then that's it for today. So once you have a drug that you've identified, that you've worked on, maybe you've tweaked, right, and you're very excited because it works really well in vitro, it kills cancer cells that represent like the cancer type that you're interested in. You show that it doesn't have really bad toxic side effects. Then you go ahead and you do experiments in mice, right? And you're thrilled, right? Because not only does it seem to actually work in the mouse to shrink tumors, but it also doesn't just kill the mouse, right? Then the question becomes, okay, now we're ready to make that big step into people, right? Which is a huge step. That's why now clinical trials are done in 3 phases, right? Designed to balance the value of what information you get out of it, while at the same time being very cautious, right? That you're not going to raise all kinds of ethical issues in the treatment of these patients with this experimental drug. So phase 1 is simply to determine the tolerance to the drug. That's all it is, right? This is where you have a large group of people in a trial, and you just give them different doses of this drug and see how they do. Not how their tumor does, right? But just, are they okay? Do their bodies tolerate having this drug in them? Right? You can generate a graph that looks like this. For example, let's say this is the increasing drug dose on the x-axis, right? And just to start off with, if it's just a simple line going across, at some point, there's a maximum tolerated dose of the drug, right, per body size of a patient who's part of this study, right? Beyond that, they have bad side effects, right? So you say, okay, we're not going to go over this maximum tolerated dose. Then you can start looking, right, at phase 2. You can start looking at how effective this drug is within that dose, underneath that dose, right, of actually treating tumors. And by doing so, by staying under that maximum tolerated dose, you can find what minimum dose is necessary in order for you to actually see some desirable effect against the tumor. So that window then, boundaried by minimum effective dose and maximum tolerated dose, is your therapeutic window, right? So from here on in, now that if you do more experiments in clinical trials, you're just playing within this range of this therapeutic window, okay? And so it's balancing, right? The blue line, which is efficacy, in other words, how well does the drug work to target the tumor, against toxicity, the pink line, right? Which is basically, are we poisoning the patient? So then the third phase is the phase 3, right? So this is the very exciting one. I'll tell you right off the bat that every year there are hundreds and hundreds of papers published talking about a new drug, a new type of molecular shape, right, that shows efficacy in cell culture and in animal models. Only about 1 in 100 of such drugs ever make it even to a phase 1 clinical trial, right? Let alone a phase 2 or a phase 3, right? By the time you're talking about phase 3, right, you are talking about that very rare drug that has made its way through many years of laboratory work, clinical trial phase 1, phase 2. Now phase 3 is where you actually establish, right, what type of cancer specifically, right, is this drug going to have as its on-label application, right, so that it can now be used particularly for that drug. So you establish the significance of the drug treatment. And this is when you have a very large clinical trial now, right? Thousands of people who have a particular disease, for example, pancreatic carcinoma, right? And what you want to do is you want to have one group receive your new exciting drug, in this particular case, gemcitabine, right? Versus another group receives the traditional popular, old-fashioned drug, right? In this case, 5-fluorouracil, that's been around for decades. And then you go ahead, you use the knowledge that you gained from your phase 1 and phase 2 clinical trials to know what dosage is good, right, to give these patients of the gemcitabine. Right? And you go ahead and you do it in thousands of cases and controls. And you have the two cohorts of the two different drugs. And this is the data that you get, right? So you can see the traditional 5-fluorouracil. This is a Kaplan-Meier plot. It's only fitting that we end our semester looking at a Kaplan-Meier plot, right? Patients' survival over 20 months that they were followed after they were enrolled and began this treatment. You can see this is the performance of the 5-fluorouracil. This is the performance of the gemcitabine, right? So it extends on average, right, the survival time of these patients by what? Maybe like 2 months, 3 months. And you might say, that's it? All of that work to discover a brand new novel drug, and it ends up extending the typical patient lifespan by 3 to 5 months, right? I'll tell you this, the FDA, the Food and Drug Administration, if they see data like this, they'll approve this drug, right? Even though it only increases survival by a handful of months. Why? Because, if you're dealing with a terminal patient who you really feel like is not going to recover from this, then at least buying them 3 to 5 extra months of life to kind of wrap up things, spend time with their family, right? That is really important. Right? But the other take-home message is this, right? This is about as good as we are these days at designing new drugs with all that knowledge, all that fancy strategy, right? This is the best we can do. So very clearly, we have to have better ways of rationally designing drugs. And all of that kind of is based upon our knowledge of the etiology, the biology of where these cancers come from, right? And that's hopefully something that I've impressed upon you this semester, right? About how complex cancer is, and therefore how complex cancer can be to treat. Right? But hopefully this also inspires some of you who might want to go on and be oncologists and try to design new drugs, right? That these are the types of challenges that you'll face, but these are the types of challenges that hopefully you can overcome, right? And come out with the next big drug to treat a particular cancer. Right? All right, okay, we'll go ahead and stop.